drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Vieira-Potter, thank you very much for joining me.
Of course, thank you for having me.
Do you want to start off by just telling everyone a little bit about who you are and what your lab does?
Sure. My expertise is in nutrition. So I have degrees in nutrition from the University of New Hampshire. That's where I did my master's degree. And I have my PhD from the University of Illinois, Urbana-Champaign. I did my postdoctoral work at Tufts in Boston in the Obesity and Metabolism lab. And my lab, we call ourselves the Fit Fat Lab. We study mostly fat cells. Most of what I do is preclinical work in rodents, but I've got lots of collaborations with some colleagues who do more clinical work, and we do an analyses of adipose samples in the lab pretty routinely as well. But most of what I do is in mice and rats. I have a little bit of cell culture work. Historically, I've done a little bit of clinical work. My work, I will say, so the most sort of important thing is my work really straddles nutrition and exercise physiology. In fact, my degrees are in nutrition. I did my doctorate with an exercise immunologist, and that's where I really got interested in exercise. And so along the way, I've also become really interested in estrogen signaling. So when I tell people what I do, I essentially say that I study how estrogen and exercise affect fat cells. So in a nutshell, that's what our lab does.
Yeah, and given that you do work in nutrition that straddles basic research, animal research on the one side, and then you collaborate with people doing clinical work on the other, and just given sort of the nature of this area of research, but also just the general chatter around it. Mm-hmm. how do you think about how to think about the relationship between basic animal research and clinical human research? Can you just give us a sense for like how do you as a researcher like look at results in your lab from animals and use that to think about the clinical side in humans and vice versa? Because some people get really you might say chauvinistic about their area of research. Some people are kind of like really clinically oriented and they say, we don't really need to look at animal research at all because it's rats and mice, they're not humans. You get the basic researchers who don't really think about the clinical side at all because, you know, they have this sort of sense of purity or whatever. So how do you sort of straddle the two sides of this?
Yeah, and it was a perfect question. In fact, I think we need all of the research. So my lab mostly does clinical, but I will say historically, when I was a just starting out graduate student, when I sat with my PhD advisor, the first conversation we had was me saying, I want nothing to do with preclinical work. I don't want to do dice mice. Clinical work is really all that I'm interested in. And he looked at me and he said, well, if you want to be in my lab, you need to do both, because I think it's important that, you know, to answer complex research questions, we really need all models. And so I sort of like hung my hat, was like, okay, well, I'm in, sure, I'll do it. And then as I got into doing more preclinical work, I kind of just went down that path, mostly because it's just sort of the nature of how things went. But I do think, you know, I don't like to tell people, what do I do for work? Well, I'm ridding the world of fat mice, right? By the time I retire, there'll be no more fat mice in the world. Nobody cares about that, right? So I use the animal model in a way that I think we cannot use the human model. And I'm someone who only uses the animal model when it's necessary. I do a lot of work that is focused on the relationship between a specific brain pathway and fat cells. And that is something you really can't do in a human. You can't take brains and look at the nucleus accumbens and look at what genes are turning on and off with estrogen exposure. That is something unique to the rodent, right? That we can do that in an animal model. We can't do that in a human. By the same token, I only address questions in the rodent that I think can translate to humans in some way. For example, one of our major discoveries over the past several years has been this really strong relationship between an estrogen receptor and a protein called UCP1, which stands for Uncoupling Protein 1, which is a mitochondrial protein that induces energy, essentially energy wasting. It uncouples oxidative phosphorylation from ATP production in mitochondria, and it basically burns off heat and it increases energy expenditure in fat cells. Well, we found this really close relationship. I mean, when you look at the correlation, it's like 0.7, 0.8. It's a really strong correlation. And I got really excited about that when I saw it in female mice. Oh, I saw it in male mice too. And then I went to my rat models. But then I had samples from a clinical study. And of course, I quickly wanted to look at in obese males and females in this clinical study, were we seeing the same relationship? And we are. So now I'm excited about that. Like, so it takes me like, you know, drawing from, you know, the clinical work. And then if we can kind of validate that in the animal model and then take it one step further, I think that's really where the preclinical work plays an important role.
Mm-hmm. And as we talk about some of the things we'll talk about today, how does the concept of evolutionary conservation and thinking about sort of the family tree of animals play into this? So when we talk about things like adipose tissue, estrogen receptors, uncoupling protein, all of these components of the biology, these things are presumably found in both the rodents and the humans, and there's a pretty good degree of evolutionary conservation there, which, and can you just say something about that and how the degree of conservation relates to our ability to translate and think across the basic research and the clinical research?
Yeah, for sure. I mean, I think the important proteins that are kind of driving biology, you think about things like mTOR, you know, estrogen receptors, those really critically important proteins are conserved. If you have a protein that's, you know, not conserved, it's not that it's not important, but it's probably not a key protein that's going to be a good you know, focus for the animal model in terms of translating it to humans. So I do think there's a lot of conservation. We're not all that different from from mice and rats, as much as we'd like to believe that we are very distinct and, much more complicated or complex than those lower organisms, if you will. But I think there's a lot of conservation, and we can capitalize on that for science.
Yeah. And so, one of the things we have in common with rodents is that we're mammals, right? We've got males and females. We've got a lot of the same hormones and stuff. And of course, we've got fat tissue. We've got adipose tissue. Before we get into some of the specific topics we'll get into, I just want to ask you to help me paint a picture for people of adipose tissue itself. So adipose tissue is fat tissue. What is it? Can you just talk a little bit about it at a high level in terms of its architecture, and what about it makes it good for energy storage?
Yeah. So adipose tissue comes in two major flavors, right? So there's white fat and brown fat. White fat is most of the fat that we have in our body, 98% probably, give or take, a few percentage points depending on the individual. But the vast majority of fat we have is called white fat or white adipose tissue. And its major function is to store energy in the form of triglyceride. And under the microscope, a fat cell, I have a picture of a fat cell in my background, but it's essentially a cell where its almost entire cell body is made up by a lipid droplet. There's just one big honking lipid droplet that takes up most of the cell. But the adipocyte cell itself is like any other cell in your body in that it has all of the standard working parts of the cell, right? Still there's nucleus and mitochondria and Golgi and all the things. They're just squished to the side because most of the cell body is taken up by this big lipid body, which is how we store lipid in fat cells. So that's white fat. Brown fat, again, the fat that we have much less of, but serves a very different purpose. It's really less focused on storing lipid or storing energy, but rather metabolizes fatty acids to produce energy and produces energy in the form of heat. And so brown fat is really important in animals that hibernate, for example. It allows for body temperature regulation. But in humans, it turns out that it's a really beneficial type of fat in that it does increase energy expenditure. It has glucose clearing abilities, and it tends to track with metabolic health. So individuals who have more brown fat tend to have less obesity, tend to have less diabetes. It tends to track, and we lose it as we age. So all of the things that you think of go down in aging, we use a little bit of brown fat. The reason I bring it up is that white fat, and so we used to just really think of fat as being white fat. And so the brown fat has only entered the discussion in the past a couple of decades or so. This is really sort of a newer thing that we're talking about more in terms of human metabolism. But anyway, so it is metabolically beneficial. It generates heat, and white fat with certain dietary and exercise practices can actually become more like brown fat. And this is sort of turning things around a little bit, because we used to think of white fat as really not burning any energy, right? We think of what tissues in our body are burning energy, are like making the bulk of our basal metabolic rate, contributing to the energy that we expend each day. Adipose tissue contributes very little, a little bit, but not much. But we can actually increase the energy expenditure of adipose tissue by increasing its browning. And what do I mean by increasing browning? Well, essentially increasing the mitochondrial content of white fat. And you can do that with things like exercise. There are a variety of things from foods that we eat, like capsaicin, which is in red peppers, can increase it a little bit. Green tea can increase it a little. So there's things in the diet you can do. But essentially, so to not belabor your question, we have white fat, we have brown fat. Now we have kind of a new fat. They call it beige fat or bright fat, where you can get white fat to act a little bit more like brown fat, which is extremely metabolically healthy. So other than those two distinctions, where fat is located on the body is also really important metabolically. So
hold on. Let's stop there for a sec.
Sure.
So we've got white fat and brown fat. White fat is probably what people see and think of most when we think about fat tissue. When we think about things like obesity, people getting bigger over time, that's mostly white fat. These are the adipocytes, or the fat cells that store fat. So literally, as you said, These are, approximately spherical cells, and they have all the normal stuff that other cells have. They have a nucleus with DNA, they've got ribosomes, they've got proteins and stuff, but most of the space in that cell is literally a big blob of fat. And it's just storing that fat, which is energy for later in case we need it.
That's right.
Okay. Yeah. So we got white fat, we got brown fat. Brown fat does other stuff. We'll probably get into that a little bit. But then there's another sort of dimension we can think along here, which is where in the body that fat is. And so, say what you're going to say there.
Yeah. So it turns out that most of the fat in the body is called subcutaneous fat. It's under the skin. And that fat serves lots of functions. But it tends to be more insulin sensitive than another type of fat, which is visceral fat, the type of fat that gets stored sort of within our internal organs, and in particular in the central region of the body. When individuals age, they begin to generate more belly fat. If you think about sexes and how men and women, males and females, store fat differently, you'll notice that most of the time females store fat with what we call a gynoid fat distribution pattern, meaning below the waist, the hips and the thighs is really generally where most of that subcutaneous fat is, most of that white fat is stored. Whereas males tend to, when they gain weight, they gain it in the midsection, in the belly area, and that's abdominal fat. The reason that can be less metabolically healthy or more kind of conducive to metabolic dysfunction is that the fat stored in that area of the body, kind of really close to the organs, tends to associate very strongly with liver fat, with hepatic steatosis, and tends to be less insulin sensitive. Mm-hmm. And so subcutaneous fat is very insulin sensitive. So example, we eat a meal, insulin goes up. Insulin's job is to get our cells to take up that energy and store it. And so fat cells are very good at responding to insulin. And when insulin is available, energy is available, fat stores take up that energy and store it. What fat cells also do, and what insulin also does to the fat cell, is it inhibits lipolysis. So when we're out on a run or we've been fasting, we want our adipocytes to be mobilizing lipids so that our skeletal muscle and our heart muscle and all of the other cells in our body can utilize that energy. But when the adipocyte becomes resistant to the effects of insulin, then insulin is not regulating lipolysis as well. And what you end up with is fatty acids being mobilized in times when the body doesn't need those fatty acids. And it turns out that fatty acids circulating around in the body is not good metabolically. Just like having high glucose in the blood is not good. Having chronically elevated levels of NEFAs or non-esterified fatty acids, which come from adipose tissue, also not healthy.
Got it. So is it fair to say, if you want to store energy if you want to store energy, you want to put it in the form of fat, triglycerides inside of fat cells for storage, or you might want to use some of those fatty acids for burning to oxidize them in the mitochondria for energy, but you don't want sort of other things. You don't want the fatty acids to sort of get out of the fat and just leak into the bloodstream and not get burned for energy and not be stored and just circulate around. Just like you don't want to have too much glucose circulating in your blood and not to either use it as a fuel source or transform it for energy storage purposes.
That's right. That's right. And part of the problem with what you just described is when you have elevated levels of fatty acids in the blood, again, it's not healthy. It wreaks havoc on the blood vessels, and they're inflammatory and they cause oxidative stress and all of the things. So what happens is they get ectopically stored, meaning they get stored in places other than the adipose tissue, which is the adipose tissue is where they should be stored. But they get stored in skeletal muscle cells. They get stored in the liver. And that really is what exacerbates metabolic dysfunction, is when lipid begins to be stored in these other organs. So having very healthy insulin sensitive adipose tissue is actually a good thing metabolically.
Right. It's almost like hoarding behavior in a human or something. Like I'm thinking of this because I'm cleaning out one of my closets that has accumulated as much as it can hold, and things are starting to leak out. So I know I need to put this somewhere else because it's not good for the orderliness of the apartment.
Great analogy.
Okay, so fat cells are adipocytes. Most of the fat cells inside of us are going to be white adipose tissue, not brown adipose tissue. These are primarily for fat storage. You've got subcutaneous and visceral fat. Subcutaneous means under the skin. Whenever I look at a little baby, I think it's highly visible. You can see that it's like they've got fat right under their skin. It's like they've got these chubby little arms and stuff. That's subcutaneous fat. That fat tends to be more insulin sensitive and less problematic in terms of metabolic dysfunction, as you mentioned, than visceral fat, the fat in our viscera in between our organs that can accumulate. That would be like beer belly fat. Is that what that is?
Right. Yeah.
Okay. So different types of fat, they have different distributions in different people. Men and women are systematically different. When people get fatter, when they become obese and they start putting on weight in the form of more body fat, what is happening there in the adipose tissue, the white adipose tissue? Is that mostly more fat cells forming? Do the cells get bigger and just filled with bigger sacks of fat? What is going on there when that extra mass is coming on? How much of it is more cells? How much of it is the cells actually just filling up with more and more stuff?
Yeah. So great question. We think the thinking used to be that you have a set number of fat cells when you're born, and that's your number, and they're going to grow and they're going to shrink, but you're really not going to get more or fewer fat cells. Turns out that that's not correct. It turns out that you do see hyperplasia, which is more fat cells, proliferation of fat cells, as well as you do see hypertrophy of fat cells, which you described as they're just getting bigger. What mostly happens with the progression of obesity is the latter. Fat cells get bigger. And the problem with fat cells getting bigger in the time course of obesity is that once they get too big, the blood supply that's going through adipose tissue isn't sufficient to fuel those fat cells. And so what happens is they essentially get hypoxic and they could die. And in a morbid obesity situation, they do. You have lots of death that's going on in the fat cells because of that. And why is that a problem? Well, what's a problem there is now inflammatory cells need to come in to clean up those dead cells. And when that happens, you get inflammatory cells like macrophages, which are, it's big eater, right? That's the big eater cell of the immune cells. So the macrophages come in essentially to eat that lipid, eat those dead cells, which is great. It's a part of the remodeling process of the adipose tissue.
Right. But now you've got even more stuff trying to go into the space, which is already too filled and constrained.
Exactly. And what happens there, it's sort of a vicious cycle because that causes inflammation because these macrophages that are there to eat the dead cells are producing cytokines, which leak out into the blood, increase inflammation in the circulation, promote cardiovascular disease, et cetera, et cetera. And what's really important in terms of the adipose tissue with those inflammatory cells is that those inflammatory cells are directly affecting insulin signaling pathways, meaning we talked about how insulin is really important in allowing for efficient storage in fat and regulating lipolysis, right? Regulating how much lipid is getting out of the fat. Well, inflammation causes insulin resistance. So you now have a fat cell that isn't as good at taking up lipids. So what happens to that lipid is it goes to other places. It goes to the liver, it goes to the muscle, et cetera. And you've got a fat cell that, again, isn't sensitive to insulin. So you've got dysregulated lipolysis. You've got fat cells mobilizing their lipid at times when they shouldn't be mobilizing their lipid. So there's all sorts of stuff that's happening in the adipose tissue as these adipocytes get too large, that mostly it causes inflammation. And my lab has become really interested in others in not only the inflammation that's happening there, but what's happening to the mitochondria. Because again, there's mitochondria and fat cells. There aren't a lot of mitochondria and fat cells. We don't think of them as being a highly metabolic organ, but they are there and they do serve a purpose. And inflammation adversely affects the function of them. And so now you've got all this lipid and you've got mitochondria, they're metabolizing lipid, and the mitochondria are not as functional as they should be. So what's happening there is the mitochondria are producing more reactive oxygen species, which causes more inflammation, which exacerbates the insulin resistance. And so I like to call the adipocyte triad. So I classify adipocyte health as the triad of adipocyte health, which is insulin resistance or insulin sensitivity, inflammation, and mitochondrial function. And I think those three things are the cornerstones of what keeps fat cells healthy. And adversely affecting any one of those three things is going to adversely affect the health of the whole tissue, which I think is part of the reason why brown fat is so metabolically healthy, is that brown fat, unlike white fat, is chock full of mitochondria. In fact, that's why it's called brown fat, because it's very dense with mitochondria, doesn't have that big one lipid droplet, has lots of lipid, but there's tiny little lipid droplets dispersed throughout the brown fat. And that lipid is being actively metabolized by all of the mitochondria in that tissue. So it's a good kind of parallel to think about, well, one fat is metabolically healthy and the other is extremely metabolically healthy. And the difference might be the functionality and the content of the mitochondria. It's a big part of it.
Got it. So just to reiterate, the whiteness and the brownness of white fat and brown fat cells is largely to do with the mitochondrial density of those cells, which is to say the white adipose tissue, the white adipocytes, the white fat cells don't have very many mitochondria because they're not doing a whole lot of burning. They have to do some. Obviously, they're cells. They've got to use energy and they've got to use mitochondria to do oxidative phosphorylation, but they're primarily a storage cell. The brown adipose tissue is brown because it has a high density of mitochondria, because it's not for storing as much fat as can be stored. It's for utilizing that fat to generate heat, as you said before.
That's right.
Interesting. So when we think about adipocytes filling up with fat, at a high level, how is the adipocyte sort of sensing or figuring out or communicating with its neighbors in terms of how full it is and when it's too full? And like how does that start to happen? Is a component of this literally like biomechanical sensation? Like there's, mechanoreceptors and stuff that physically sort of feel the pressure within the adipose tissue and how full these cells are with fat. How do these cells know when to start communicating, like, hey, we've got room for more, or hey, we're totally full and we've got to do something about this?
Well, that's a really, really interesting question. I don't know about the mechanoreceptors and sort of like the mechanisms that the fat cell goes through to kind of make these decisions. I really think in terms of insulin signaling and really how the lipids are getting in there. And most of it, like and it's not that I just don't know that side of it. I've really not thought too much about it, but it's a really beautiful question.
But let's talk about how the fats actually get in there. Give us a high level overview of that process. So obviously, we're eating food. Some of that food is fat, and we've got more than enough. So our body's going to just store it for later. That's what the whole purpose of the white adipose tissue is. How do those fatty acids actually get imported into these cells? Yeah.
So there are fat. So there are enzymes on capillary cells that sort of line the adipocyte, and there's an enzyme called lipoprotein lipase, and lipoprotein lipase is the enzyme that allows that fat or those fatty acids to enter into the adipocyte. So lipoprotein lipase is an important enzyme there, and insulin acts on that enzyme. So insulin sort of is the key that opens that lock that allows the adipocytes to get into the adipocyte. And there's actually a whole branch of science on the fat cell biology and the lipid droplet biology, because there's a lot that's going on at the level of the lipid droplet. So there are proteins that sort of surround the lipid body in the fat cell that allow for lipids to enter. And that's not my area, although I did my postdoc with Andy Greenberg at Tufts at the Obesity Metabolism Lab at the USDA Nutrition Center. And what he did when he was a postdoc was he was part of the team that discovered a protein called perilipin, which is a really important protein that allows for the fat entry and exit from the lipid droplet.
Mm-hmm.
So essentially it's insulin allowing fatty acids to enter, and then when the fatty acids are in the adipocyte, fat metabolism in the adipocyte is very similar to fat metabolism in other cells of the body, like skeletal muscle cells, in terms of, the fats get into the mitochondria via specific proteins called CPT1 and CPT2 are enzymes that let the fat cells into the mitochondria. And I always tell my students, like, the mitochondria is like a furnace. Once fat cells get in there, they get oxidized. So there's regulation at the level of fat cells getting or fatty acids getting into the mitochondria, but that's kind of where the level of regulation is.
Got it. So there's various proteins and enzymes that help import fats into adipocytes, and the hormone insulin is plugging right into those mechanisms.
That's right.
Got it.
That's right.
So one more question that may or may not be exactly in your wheelhouse before we get to some of the work that your lab does, but I want to ask you about it because I'm curious about this general area. How much does the fatty acid composition of the fat stored in adipocytes start to affect things like insulin resistance and inflammation? So for example, if you've got a difference in the relative abundance of saturated fatty acids versus highly unsaturated fatty acids, how does that play into, say, their sensitivity to macrophage infiltration, oxidative stress, and those types of things?
Yep. I don't study that directly, but I will say that it absolutely plays a role. And, you know, a triglyceride is a glycerol backbone with 3 fatty acids attached to it. And there are, you know, a heterogeneous assortment of fatty acids that might enter into that triglyceride. And you're going to have saturated, you're going to have polyunsaturated, monounsaturated. Saturated fats act very differently than polyunsaturated fats. Saturated fats are solid at room temperature. They're more rigid. Polyunsaturated fats are more fluid. And so as far as insulin signaling and any type of cell signaling goes, more unsaturated fatty acids are going to be conducive in general to better signaling, just because it allows the membranes to be more fluid and receptive to signaling pathways and all of that. Whereas, you know, a high density of saturated fats will just make things a little bit more rigid, and that can adversely affect signaling pathways. Saturated fats also tend to be more inflammatory.
And when you're saying signaling, people might imagine the cell membrane, and there's proteins in the membrane, there's ion channels, there's transporter proteins, there's GPCRs, there's a bunch of proteins in the membrane. They're not sitting statically. The whole thing is like sort of literally fluid. So there's motion of these things, and the ease with which proteins can move through the membrane affects the rate and the quality of the signals being communicated inside and out of the cell.
Yeah, absolutely.
Is that fair? Yeah.
Yes.
And so, okay, so there's a difference between unsaturated and saturated fatty acids. Saturated fats are saturated. They don't have double bonds. They will tend to decrease membrane fluidity and make things a little bit more, it's like think of a fat that's solid at room temperature. Unsaturated fatty acids increase membrane fluidity. So things will move around a little bit more. We don't necessarily need to dig into all the details here. I'm assuming there's lots of sweet spots that are better or worse for different aspects of signaling and cell biology, where you might want to increase fluidity or decrease fluidity depending on circumstance. But you're saying that the balance of these fatty acids, saturated, unsaturated, affects membrane fluidity, and that has some impact on signaling and how well it's happening.
Yeah, I think that's accurate.
Yeah. What about the fat that's actually stored inside of the cytoplasm? Is there a tendency to store saturated fat more than unsaturated fat, or vice versa? Like, is one of them better for like storage purposes, or?
Yeah. So again, triglycerides are sort of heterogeneous in terms of the types of fatty acids that are within them. I think one thing that is important is our bodies make fat, and the fat that our bodies make is called palmitate, and it's a saturated fat. So when we de novo lipogenesis, or making new fat from sugar, essentially, happens in the adipose tissue. It also happens in the liver. And so the bulk of the fat that's stored in the adipose tissue is probably saturated fat, just by the nature of that's what's produced there. The fat cells are very good at, taking up fat and storing it. And it doesn't really matter. The fat cell doesn't care what type of fat it is. It'll store it as triglyceride in that lipid droplet. But the fat that it's making is palmitate.
Yeah. Okay. That's interesting. So that's worth highlighting for people. So there's many ways to be in a state of caloric surplus. You can eat a lot of sugar. You can eat a lot of fat. It might be saturated fat. It might be unsaturated fat. There's lots of ways you can eat way more than your body needs to burn. And in all of those cases, it will store some of that excess in adipocytes. So if you were let's say you were eating caloric surplus, but it was like pure glucose, pure carbohydrate, you're not consuming any fat. Well, you would still put on body fat, and that's because you're going to stimulate these adipocytes to make palmitate, a saturated fatty acid, and that's what will fill them up. Conversely, you might eat a lot of fat, and maybe it's some other cocktail of fatty acids. And in that case, you might stimulate some palmitate synthesis in the adipocytes, but also you might store some of those fats that you're eating. And then the fatty acid composition of what's in the fat will start to reflect, to some extent, the composition of the fat that is in your diet.
Yeah, that's accurate.
Interesting. Okay, so palmitate, a saturated fatty acid, is the one that our bodies actually synthesize. I guess that's why we say that unsaturated fats, like polyunsaturated fats, are essential, meaning we need to acquire them for a diet. Our body can't actually make them.
Yeah. Well, it's actually, so our body makes saturated fat, it makes palmitate. There are desaturase and saturase enzymes. So we have enzymes that we can desaturate, or we can elongate, elongases. So we can elongate the palmitate, we can desaturate. However, we do lack the enzymes to make essential fatty acids. So DHA and EPA are essential fatty acids, linoleic acid, linolenic acid. Those fatty acids we do need in our diet, because our body doesn't have the machinery to make them. So we start with palmitate, and then we can kind of tweak it and make some fat, some unsaturated fats, but certainly not all of them. So you're absolutely correct that there are essential ones.
And I've covered this on the podcast before and in my writing, but what people can imagine here is, okay, so you make palmitate. That's sort of like, I don't know, that's the baseline fat that the cell makes. But then there's enzymes that can make it longer or add a double bond, but there's only so many double bonds that can be added, and only in certain positions. So we can make something like a monounsaturated fatty acid out of palmitate, but we can't make something like EPA, like fish oil fat from that.
That's right. We cannot make fish oil, which is why fish oil it contains essential fatty acids, and need that in the diet.
So we touched on it briefly, and this is pretty obvious to most people from experience, but one of the interesting things about body fat in animals is that it changes over the course of development. And then there's also systematic differences between males and females. And so I want to talk a little bit about that. And the tie-in here that I think is really interesting that gets into your work is the sensitivity of adipocytes and adipose tissue to hormones like estrogens. So let's give people a lay of the land here. What is estradiol and what are estrogens? Let's give people a basic definition and talk a little bit about sort of their chemical properties in terms of the fat solubility, and also the diversity here. How many estrogens are there? Are there a lot, or are there just a few?
Okay. So when we think estrogen, we think the major female sex hormone, right? Females generally have estrogen as their major sex hormone. Males have testosterone as their major sex hormone. And you're right, estrogen is kind of a group of molecules. The feminizing type of estrogen, the estrogen that we think of when we think of, the major female sex hormone that allows for reproduction and all that, that's 17 beta estradiol. And that is the major form of estrogen. There are also, there's actually another form called 17 alpha estradiol. That's a less prevalent form of estrogen. We also have estriol. We have a specific estrogen in pregnancy, but those are the main ones. There's a few different types of estrogen, and then there's actually, so that's the steroid hormones. The reproductive steroid hormones, but we also have plant estrogens. Well, we don't. I mean, plants also make estrogens, like for example, in soy products, there are phytoestrogens, and they look very similar to the estrogens that I'm describing to you, but they bind to estrogen receptors with much less affinity, and so they do not have the effects that the natural steroid estrogens have in physiology. But estradiol is 17 beta estradiol is the major estrogen that is, So So
there's multiple estrogens, but estradiol is the one we talk about the most. It's probably what people think are actually referring to when they say estrogen, even though estrogens are a family of compounds. There's estrogens our body produces. There's plant estrogens like phytoestrogens, like genistein and others that we can talk about in other contexts. But these are hormones. And so what does that mean in terms of, so just abstracting this from the fat tissue stuff for a moment, how do estrogens like estradiol have effects on cells? What does the signaling pathway there look like? So
the most important part of this is that they bind to receptors. In the estrogen receptor, so there's there's three major types of the estrogen receptor. Mm-hmm. There's estrogen receptor alpha, estrogen receptor beta. Those are nuclear transcription factor estrogen receptors. And then there's a GPERS, a G protein coupled estrogen receptor. So those are the three main types. And the important thing to know about the estrogen receptors is estrogen doesn't do anything except through its receptors, mm-hmm. right? These estrogen receptors are all over the body. And by the way, males and females both produce estrogen. I should have mentioned that. And both males and females produce testosterone. It's just that the ratios are different. And obviously, estrogen is the major sex hormone in females and female reproduction. But males and females both have estrogen. Estrogen receptors are widely dispersed throughout the body, the brain, the skeletal muscle, the adipose tissue. Most cells express some level of estrogen receptors. So estrogen affects cells by binding to its receptors. And then it's a little bit, so the estrogen receptors are a complicated receptor because they're a steroid nuclear transcription factor. So meaning they bind to estrogen and they can actually go to the nucleus of the cell and change gene expression.
Yeah.
So this is a really important function of estrogen. But estrogen receptors are also in the cell membrane. So they also mediate rapid signaling effects. So there are genomic effects, which are the effects that estrogen regulates at the level of the nucleus of the cell, regulating genes to be up and down regulated. And then there are non-genomic, or we say rapid signaling effects of estrogen, which involve estrogen binding to the receptor on the surface of the cell.
Mm-hmm, mm-hmm. So 3 estrogen receptors. Any given cell might have all 3 or 1 or 2 or some combination of these, but many cells are sensitive to estrogen through some combination of those receptors. There is a membrane estrogen receptor on the cell membrane that estrogen can bind to switch on signaling pathways in the cell. But there's also estrogen receptors that are inside the nucleus. So estrogen can get into the cell, into the nucleus of the cell. And when it binds to that receptor, that's a protein that's then binding directly to DNA to flip genes on and off.
That's right. It's actually called a nuclear transcription factor because it binds to the DNA in such a way that it can up and down regulate groups of genes at the same time. So it's a really powerful regulator of gene expression.
Right. So when estrogen binds to one of these things inside the nucleus, it's not just flipping on a gene or flipping off a gene. It's actually flipping on combinations of genes on and off. And so this might get people thinking about, like, how is it that hormones can powerfully orchestrate developmental shifts like puberty or menopause, say, where you have a complete rearrangement of tissue systems into new things that do stuff that wasn't there before. It's because these hormones get right into the nucleus and they're completely changing gene programs that are then going to ultimately influence what cells are there, how many are there, and how they behave.
That's right.
And so, estrogens are in males and females. We all have all of these components. It's just, the relative ratios of these things differ. I kind of guess maybe it would be instructive to kind of walk through some of the major developmental stages in females, mostly, to understand the effect of estrogen, in particular, on fat tissue. So one place to start maybe is puberty in females. One of the hallmarks of that transition is that the adipose tissue changes. The amount of fat and where it's distributed on the body changes. Can you maybe overview that for people a little bit? What are the major changes in the adipose tissue that are happening to females at the onset and throughout puberty? And how is estradiol actually orchestrating this and starting to change the adipose tissue?
Yeah, well, I think the fact that one of the major things that we see happen during puberty is a difference in adiposity, right? And how we store fat and the amount of fat that's stored. This is something I think that happens in development, particularly in females, but I think in both sexes. And I think that is a good example of how important estrogen and the sex hormones are in forming adipose tissue and kind of in directing kind of where it goes and how it's distributed in the body. Actually, I don't know if this is a tangent or not, but a side note that I think is important is there is a hormone that's produced in adipocytes called leptin. And this is a really important hormone. It was discovered in the 90s by a guy named Jeff Friedman is a really, really important discovery, because what he showed with leptin is that it's a hormone that is released from the adipose tissue, but it regulates energy balance. So it binds to receptors in the hypothalamus, and it allows for the adipose tissue to regulate energy storage. And so when the adipocyte gets too full, leptin can signal this in the hypothalamus region of the brain, to increase satiety, right? And so there's this really nice mechanism that starts in the adipose tissue. And the reason I bring it up with your question on puberty and adipose tissue development is that individuals, and there aren't many individuals with this genetic mutation, but there are some, a very small number of individuals are born with a genetic mutation in the gene that encodes leptin. Mm-hmm. Those individuals do not reach sexual maturity because leptin is so critical. Females going through puberty will not, for example, females going through puberty will not start menarche until they have a critical threshold of adipose tissue. Okay. It's necessary for puberty to happen. And that is true in males and in females. Got it. And it's because of leptin. That's one of the things. I'm sure it's more complicated than that. But.
So leptin is often referred to as a satiety hormone. That's maybe a little bit simplistic, but it's a very important metabolic regulator, among other things. It's important maybe for people to know, okay, so it's synthesized in fat tissue. So that's right. The health of your fat tissue, the amount of your fat tissue is going to matter for how much leptin is made and how much ultimately goes up to the brain to do its thing there. But also the amount of fat tissue a female has, that males or females have, but that females have as they go through puberty is going to be tied to, I would imagine that the way to start to think about this here is if it's tied to menarche, that starts to make sense if you start to think about it from the perspective of what puberty is setting up the body to do. The female ultimately will be set up to be able to give birth to a child, and fat's going to be very important for that, because that fat tissue is going to need to be filled with the energy that's going to help nourish that child.
Yeah, you got it. Exactly. The other thing I will mention, which kind of most people don't appreciate this in terms of what's going on in fat cells, but fat cells actually produce estrogen. And part of the reason why girls who are developing obesity, young girls who are developing obesity, when they're sort of in that going through puberty phase, will start menarche earlier. Right. I mean, girls are starting earlier, 7, 8, 9 years old. Reason is that the increase in adiposity is actually, increasing more estrogen production.
Yeah. I see. I had heard before that the age of onset of puberty in females had been creeping upward, getting earlier in development for some time. But I don't know if I've ever heard this explanation. People have said, well, maybe it's endocrine disruptors, maybe it's, estrogens.
Could be a variety of things.
Could be a variety of things. But one factor that makes a lot of sense that you just said that I didn't know about is it could, to some extent, it could be an artifact of childhood obesity, because the more fat tissue you have, the more endogenous estrogen you'll produce, and that'll stimulate this phase to start earlier.
Yep, absolutely. We have an enzyme called aromatase in fat cells that actually converts testosterone to estrogen.
Yeah. Interesting. Okay. So let's just assume, puberty is happening normally. Everything's going as it should. What are some of the just describe here, what are some of the major changes in the amount and the arrangement of adipose tissue in a female? How is the distribution in the body changing, and how's the overall sort of adipose tissue, density and what the cells are doing? How is that actually changing as puberty progresses?
I think it has to do with the estrogen receptor expression. I think that's probably the biggest driver of where fat is being distributed. And I think sort of, young kids kind of emerging into the, pubescent years and going forward from there, you begin to see the body fat distribution changing. Changes in estrogen receptor expression on various cells in the body and even in various adipose tissue depots. So, for example, I mentioned estrogen receptor alpha and estrogen receptor beta. The estrogen receptor alpha is the feminizing receptor. It is the receptor that's the predominant receptor on fat cells, and females have greater expression of this receptor. And have greater expression on fat cells. And I think the the changing of where the expression levels of adipose tissue of estrogen receptors in adipose tissue is probably a major predictor of how adipose tissue is distributed in the body and how that changes through puberty.
Got it. So there's two factors here that will lead to sort of a different developmental trajectory for males and females. The overall amount of circulating hormones is different. The relative balance and the absolute amounts of estrogens and androgens is different. But also cells start to express different patterns of these different receptor subtypes. And I would imagine that different cells within females, but also when you compare males to females, they're going to be expressing different combinations. And so their cells will be differentially sensitive to these estrogens that wax and wane throughout development.
That's right. That's right. And actually, a good example of that is, I mentioned these kind of sex-specific fat distribution patterns. Well, that changes in menopause in women. And in fact, in menopause, women begin to take on a more male-like fat distribution pattern. They tend to accumulate more belly fat in less fat in the lower region of the body. And part of that is the drop in estrogen. And again, the feminizing form of the estrogen receptor is estrogen receptor alpha.
Yeah.
And the reduction in estrogen that we see in menopause is going to be followed by ultimately a suppression of that receptor. And the deposition of adipose tissue begins to change because of that, and you get more accumulation in the central region. Got it.
And we'll probably get back into this stuff more later. But so when you get to like menopause and stuff, it's not just estradiol goes down, but the expression of the receptors is also changing, which presumably is why you can't just inject more estradiol and go right back the way you were before. It's because your cells are differently sensitive to estrogen than they used to be.
That's right.
Interesting.
That's right. I mean, estrogen plays a major role. In fact, one key example of why the estrogen, it's all about the receptors. I mean, we hear so much about estrogen, but really, estrogen is not going to have any effect if it's at the level of the receptor. And so an example of how important the receptors are is that immediately following menopause, we see an increase in receptor expression because it's very important. And the body is, physiologically like trying to capture any molecule of estrogen that's circulating around, right? It's sort of adaptive. Yeah. But then, of course, as the years go by, that level drops down because, there's no estrogen. And so there'll be a downregulation of that receptor. But yeah, it's all about the receptor density.
One thing that I think could be useful to help us think about menopause stuff more later on in the discussion is what estradiol is doing within adipocytes or other cells in terms of mitochondria and metabolism. So can you talk a little bit about how estradiol affects mitochondrial biology in cells like adipose tissue that are sensitive to estrogens?
Yeah, this is one of my favorite things to talk about because I think it's fascinating. So actually, I should mention a book because there is an entire book, Power, Sex, what is it called?
Are you talking about Power, Sex, and Suicide?
Yes, that book. So there's an entire book about the relationship between estrogen and the mitochondria. So I think estrogen absolutely in all cells of the body really it is a really important regulator of mitochondrial function. Within fat cells, I think that I don't think fat cells are an exception to that. So one kind of broad example, in one of the early examples that really got me really interested in estrogen in the mitochondria, is that there was a clinical study conducted more than a decade ago now, which showed that the energy expenditure contribution of adipose tissue in premenopausal females was significantly greater than that same mass of adipose tissue from males. And so males have more lean to fat and females have more fat to lean, but the contribution of the adipose tissue to energy expenditure is greater in women. And I think it has to do with the fact that estrogen regulates mitochondria. I think there are more mitochondria in fat cells from females compared to males. I don't know if those studies have been done explicitly, but no, we do know estrogen regulates mitochondria. And again, this happens through estrogen receptors. estrogen receptors are actually embedded in the mitochondrial membrane of many cells. This has been shown in neurons and skeletal muscle cells and many cells. Right. And this is true in adipose tissue as well. And so this is something that my lab is particularly interested in. And we think that there might be estrogen receptors that are signature to or just innate to the mitochondrial membrane and just constitutively there. We also think that estrogen receptors might migrate to the mitochondrial membrane. And the other important thing to mention, I think, is that mitochondria have their own DNA. And the mitochondria are actually maternally inherited. So we get the mitochondria from the egg at conception. And it's maternally inherited. It's got its own DNA. And so all of it makes sense that estrogen is really going to affect the mitochondria. I think estrogen may even act as a mitochondrial transcription factor. So when we talked about the estrogen receptor acting as a transcription factor in the nucleus of the cell, this hasn't, I don't think, really been tested, but maybe, I'm not sure. I haven't done a look at the literature recently, but I would not be surprised if estrogen was actually acting at the level of the mitochondrial DNA to change gene expression.
Yeah. But I mean, the important thing is people need to remember, like when we think about something like just development and we think about something like puberty, hormones are changing. They're waxing and waning in complicated patterns. Estrogen levels might go up. Estradiol levels might go up in males and females. Even though we're going to be largely talking about female development stuff, and even though we're largely talking about the effect of estrogens on mitochondria and fat cells, the estrogen is going everywhere. It's going to bind to receptors on neurons, on fat cells, all sorts of different types of tissues. And this is how hormones orchestrate these sort of major changes throughout the body, is they're going through the bloodstream, getting everywhere. And this is leading to coordinated changes in gene expression across all tissues, which is why, like when puberty starts, your body physically changes, but also you start to act differently and your behavior changes because it's going into your brain and your metabolism starts to change, et cetera, et cetera, et cetera. So let's just keep that in mind in the back of our minds. But anyway, so the adipose tissue has estrogen receptors at the cell membrane, but also these nuclear receptors that can affect gene expression. So estradiol is getting onto the cell membrane. It's also getting into the nucleus of cells. It's starting to flip different things on and off. What is that doing how are the fat cells then changing as puberty onset happens and progresses? In terms of mitochondria in particular.
And that's a great question. I do not know how puberty per se affects mitochondria in fat cells. That's a great question. I love that question. I don't know the answer to it.
Okay. So that area is still unexplored.
Oh, yeah, totally unexplored. And to my knowledge, but I will say that polycystic ovarian syndrome is a syndrome that often manifests during puberty or shortly thereafter.
Yeah.
And it's a metabolic condition where females have polycystic ovaries, and a signature aspect of that syndrome is insulin resistance and mitochondrial dysfunction.
Yeah.
And it's not that they have increased estrogen or decreased estrogen. There's sort of a dysregulation of sex hormones. There's high levels of testosterone. There's all sorts of things that go on in PCOS. We're not even near understanding that syndrome. But I think it's a good example of kind of the immunometabolism and how immunometabolism of fat cells intersects with sex hormones. Because I think sex hormones and the immunometabolism, so how the insulin receptors are working and how the mitochondria are working, are all effective in many ways. And it's just, I think it's complex. And I don't know the answer to the question of how exactly are the mitochondria in fat cells changing with puberty. So our work has really been interested in looking at how does estrogen per se affect mitochondrial function and density in white cells and in fat cells. And it looks like it does. It looks like estrogen induces this, what we call browning of white fat. It increases mitochondrial activity. It increases mitochondrial function.
Got it.
density.
And so as we get into this stuff, as you study it, this is mostly in adult animals that are already sexually mature?
Yes. Yes.
Got it.
Yeah.
Okay, so
and mice.
estradiol can start to induce like a browning in adipose tissue. In other words, white adipose tissue starts to get a little more brown, which we said before means more mitochondria and therefore more mitochondrial metabolism.
That's right.
So it's stimulating like mitochondrial biogenesis and like literally an increase in the number of mitochondria?
Yeah, we think so. Yeah, that's probably fair to say. We haven't quantified numbers of mitochondria, but we look at things like citrate synthase activity in the mitochondria. We have done studies looking at specific estrogen receptors. And so we use a lot of genetic models, genetic rodent models, to tease out what is the contribution of one specific estrogen receptor to one aspect of physiology. And one of the things that we've done is we've compared mice that lack estrogen receptor beta, for example, which is the receptor that I've become particularly interested in because it's one that I think is really particularly important in the mitochondria of the fat cell. But we've looked at mice that don't express ER beta or estrogen receptor beta and mice that do. And we've looked at their mitochondrial function, meaning we have isolated out adipocytes. And then we assess using this tool called the Ouroboros, which is a way to assess mitochondrial activity. You can measure ATP production and oxygen consumption. And we show that the mice without estrogen receptor beta have a reduction in mitochondrial oxygen consumption. So there is a reduction in a significant reduction in mitochondrial activity in mice that systemically, whole body, lack estrogen receptor beta.
Got it. So just the sensitivity of cells to estrogen, whether or not they have this particular receptor, it can have big impacts on mitochondrial function.
That's right.
One thing I want to ask you about is, so before we said we've got white adipose tissue, we've got brown adipose tissue. Mm-hmm. That has to do with mitochondrial density. White adipose tissue is fat cells that are more concerned with storing fat for later, just fat storage. The brown adipose tissue, which we have much less of, is more for burning fats, oxidizing them. It's for generating heat. There's this concept in mitochondrial biology of coupling versus an uncoupling. We talk about how coupled the mitochondria are. What is that? Let's explain that so that we understand where we're going next.
Yeah. So I mentioned UCP1 earlier, which stands for uncoupling protein 1. That is the signature protein of brown fat. In fact, when we look at brown fat activation, what we can do is we take one chunk of brown fat from an animal that doesn't have brown fat activated, and one that does, and we would measure UCP1 content. And UCP1 content is our kind of marker of brown fat activation. Well, why is UCP1 the marker of brown fat activation? The reason is it's the uncoupler in the mitochondria that really allows for uncoupling of oxygen consumption and ATP production. So in other words, for set amount of oxygen that is consumed in brown fat, there's going to be less ATP produced. Some of that energy is going to be blown off as heat, essentially. So you produce heat, but you don't produce ATP, which means you need more oxygen to get the same amount of ATP.
Right. Okay. So in mitochondrial biology, they're doing oxidative phosphorylation. The reason we breathe oxygen is to get oxygen to the mitochondria. And the oxygen is sort of like pulling electrons through the mitochondria, and that's tied to ATP production. And the extent to which those two things are tied together is coupling. So if the mitochondria are very coupled, the oxygen is basically going to ATP production and doing that thing. As things become uncoupled, which they can for different reasons, there's less electron flow to make ATP. And instead, and we don't need to get into the details, instead, biophysically, heat gets created instead.
That's right.
Got it. And
That's right.
So what, like, why does uncoupling happen at all? I guess one obvious potential answer here is if we need to generate extra heat because we're in a cold environment or something like that. But like, what are some of the big factors that regulate how uncoupled the mitochondria are from coupling oxygen to ATP production?
Yeah. Well, you're right on in that the brown fat, its signature is that it is uncoupled. And so the uncoupling, because of the uncoupling proteins in the mitochondria, allow for heat production. You mentioned the cute baby earlier with having more white fat. The newborn baby actually has more brown fat as well. Reason is that the baby can't get up and get a sweater when it's cold, right? And so we're born with more brown fat as sort of a way to regulate our body temperature, and in particular to make sure that we have enough, our body temperature is high enough to protect physiology, and we kind of lose it as we age. But back to kind of why we have the uncouplers. In sort of at the cell biology sense, is there really, other than heat production, is there a reason for it? There have been some studies, and some people have hypothesized that uncoupling is a way to dissipate oxidative stress. So it might be a compensatory mechanism at the level of the mitochondria to relieve some of that kind of oxidative pressure. And that might be a way to relieve some of the oxidative stress. As mitochondria are active, they're producing the reactive oxygen species, and so the act of living, right, is killing us. We know that because our mitochondria are always producing reactive oxygen species. The nice thing is that physiology in many ways compensates for that, and we get things like natural antioxidant proteins that are upregulated. But I think uncoupling proteins might be a way to regulate that a little bit. Yeah. So possibly they have a role in cell biology that's beyond heat production.
Yeah. And I don't know much about uncoupling proteins, except that they're involved in uncoupling the phenomenon. And I understand that, but I don't understand how these things work. So how do uncoupling proteins work? I would imagine that at some level, if people imagine the mitochondria, they've got an outer and an inner membrane. The inner membrane is sort of like a wire, and electrons are flowing through it by hopping from protein to protein. They're kind of being pulled through by oxygen. Do uncoupling proteins just sort of get in the middle of that and mess it up? How do they actually do the uncoupling part?
Oh, that's a great question. And you're much better at explaining that than I am. So I don't know if I can add to that in any way. But I think I usually think of it as like creating a pore in the membrane. So they somehow uncouple, they create more of or less of a pressure gradient, which now creates a need for more oxygen to be pulled along. But I essentially think of it as a pore. Got it. In the membrane.
So instead of the electrons,
a hole in the membrane.
Yeah, it's like poking a hole in the membrane. So yeah, if we imagine a garden hose and we poke a hole in it, some of the water is going to leak out of the hose. And therefore we need more. Yeah. Okay.
Yeah, that's a good way to think about it.
Interesting. So they act in some sense literally as a pore. They're allowing those electrons to leak out.
Yeah.
Interesting.
Yeah.
Okay. So what have you guys started to find in terms of exercise and physical activity, and how it ties into this sort of connection between estradiol and uncoupling in the mitochondria?
Yeah. So one of the most interesting things I think that I've observed kind of throughout my career has been the impressive parallels between what estrogen does and what exercise does, particularly to fat cells, but I think maybe to mitochondria even more globally. So like I think I mentioned earlier that exercise actually can stimulate browning of white adipose tissue. We do know that exercise increases mitochondrial density in adipose tissue. I mean, we of course know this in skeletal muscle. We've known that with for years, but it looks like exercise also increases mitochondrial density in adipose tissue and might cause this like browning, or does cause browning of white adipose tissue, as does estrogen. So there's those parallels. I forgot what was the second part of your question.
I was just wondering if you know anything about how this. So exercise in general sounds like it increases mitochondria in cells, including adipose tissue. Does estrogen tie into that in an important way? Does it have an effect on terms of how much mitochondrial stimulation you get from exercise?
Yeah. So one of the major questions my lab is trying to answer is whether or not there is synergism between exercise and estrogen. And so the reason we're asking that question is, well, even like if you step it broadly, you things like you hear people say, that are for example, women going through menopause might say something like, I'm exercising, but I'm not losing weight. Like before menopause, I would, exercise and I would lose weight. And now I'm less responsive to exercise. And what my lab is looking at, and in the work we're focusing on, I think would suggest that has to do with the fact that estrogen does facilitate mitochondrial activity, as does exercise, and there might be relationships there. So estrogen might facilitate the responsiveness to exercise in terms of mitochondria. And so what we found was the exercise, which, as I said, increases browning of adipose tissue. It also increases estrogen receptor expression in adipose tissue. So we find that exercise increases estrogen receptor expression in adipose tissue. And we've also seen that we use a drug called CL316243. It's a beta-3 adrenergic receptor ligand, which means it's a chemical that stimulates adipocytes in much in the same way that exercise does. And that chemical stimulates browning, as does exercise, but it also increases estrogen receptor expression. And so we're trying to figure out if there is synergism there. Like if you expose cells to, or an individual to exercise and estrogen, do you get more fat loss, for example? And our research in mice, our preliminary research in mice suggests that is true.
Got it. So exercise is not just affecting the number of mitochondria you have and mitochondrial metabolism, it's also changing a cell's sensitivity to estrogens by changing expression levels of the estrogen receptor.
That's right.
And so that's rodent work. Is that in female mice, or is it in both males and females? Do you see similarities and differences there?
We actually see the same in female and male mice. I will say that in terms of sex differences, it's important to note that females are more sensitive to browning. So those are studies we did several years ago. We took male and female mice and we exposed them to this drug that I described, the CL316243, which induces browning, and showed that the females were more responsive to that drug than the males. So there is lots of anecdotal data to show that females tend to be more responsive premenopausal or estrogen sufficient females.
Mm-hmm. So basically, in rodents at least, exercise makes at least certain cells more sensitive to estradiol.
Yeah, theoretically, based on the fact that the estrogen receptor levels increase. Right.
Interesting. And so when we start to think about like later phases of development, so obviously puberty happens, you reach sexual maturity. A lot of that is instigated by changes in not just hormone levels, as we said, but also the pattern of expression of receptors. So the sensitivity of cells to those hormones, and then we go through our lives. And in long-lived species like humans and certain other animals, we then have this later phase of life that a lot of other creatures with shorter lifespans don't have. And one of the things that happens to female humans and certain other species like whales, I think, and maybe elephants, is they go through menopause. So can you let's just give people an overview again. What are the basics of menopause in terms of how hormones and hormone receptors are changing? Can you just remind us of that part?
Sure. So the main thing that happens in menopause is a rapid drop in estrogen, right? And then there's this whole phase that we're just starting to talk more about, which is perimenopause, which is it's much more elusive than the definition of menopause, because menopause is simply in a female human, 12 months without menses is diagnostic for menopause. So you classify it as menopause if you haven't had a menses in 12 months. So that's what it is. But it's essentially a wiping out of estrogen production. The ovarian estrogen production essentially stops. This is different from sexual aging in males, where there's really a much more gradual reduction in testosterone and just different changes that happen there. But menopause is really characterized by a rapid cessation of estrogen production. And that's driven by the ovarian production, basically stopping.
Is there a particular trigger for that?
Oh, that's a great question. We don't know. It's interesting that it tends to be very, in terms of the age at which menopause happens, is about age 51 in a female, but perimenopause, again, can start from, a decade earlier than that. We really don't have great definitions. There's not a lot of research at this point on this. In terms of, the physiological mechanisms that trigger menopause I think it's complex. I think probably genetics and maybe some environmental factors. But I don't think there's I think it's different depending on, there's heterogeneity in terms of when women go through menopause. But what exactly triggers it, I'm not sure.
One of the things that you wrote about in one of your papers that was interesting is it's tied to the fact that there's lots of differences between males and females in terms of disease risk profiles, the pattern of chronic metabolic disease and how that unfolds throughout life. And you wouldn't necessarily, or at least I didn't before this, connect it to things like differences in estrogen signaling. But you wrote that females are actually protected against metabolic diseases compared to age-matched males.
That's right.
Until they reach menopause. And so then they have this natural state of estrogen depletion, which basically menopause is, and that's associated with an abrupt increase in metabolic disease risk. So articulate that a little bit more for us and unpack what is the connection between estrogen deprivation or relatively low levels of estrogens and chronic metabolic disease.
Yeah. So before I was talking about the fat distribution pattern differences between men and women and how that shifts in menopause, I think that's actually a big part of it. I think estrogen, I'm fat cell centric because that's what I study. So that's kind of where I'll go with this, but I think it's broader than just fat cells. But if you even just think about fat cells and what estrogen is doing to the adipose tissue, estrogen tends to make fat cells more estrogen sensitive. More insulin sensitive. And insulin sensitivity predicts metabolic health. And so in general, females tend to be more insulin sensitive. And part of that is probably what you alluded to earlier, which is females are going to bear children and we need sufficient energy. And we need our cells to be very sensitive to insulin because we need to be very good at storing energy in a nice, safe way. And the adipose tissue is a perfect vehicle for that. So I think evolutionarily, there are reasons why females tend to be more insulin sensitive, and that extra insulin sensitivity makes weight loss a little bit trickier. If you have a premenopausal, a married couple who are trying to lose weight, the male tends to lose it easier than the female. It has to do with the fact that we're just really good at storing fat. And we're very insulin sensitive. And it's a lot driven by estrogen, as exemplified by the fact that you're right, we're protected, except after menopause, there's a risk reversal such that women are more prone to diabetes, more prone to obesity, more prone to cardiovascular disease. And the major driver of that is probably insulin sensitivity, which is why we gain more weight in the midsection. That tissue tends to be less insulin sensitive.
So the basic idea here is menopause happens. That means there's going to be a steep drop in estrogen production, and therefore many cells in the body, including adipose cells, will become more insulin insensitive. And so you're going to see a change in body fat distribution, but also you're going to see a different chronic metabolic disease profile because so many chronic metabolic diseases come with insulin resistance.
That's right. Fatty liver, for example.
Yeah. So basically, after menopause, women become more prone to things like fatty liver, which before menopause, men are probably a little bit more prone to than women.
That's right. And there's also I talk all about fat tissue, but insulin is also really important. It's a really important anabolic hormone. Mm-hmm. And as we age, we get anabolic resistance, meaning it's more difficult to hang on to our lean. And so women are at the disadvantage of starting out with less lean and more fat. And then with menopause, we kind of increase that anabolic resistance. And now we're losing even more lean. And what happens with that is the energy expenditure goes down because most of the energy expenditure is due to our lean mass. And so now you gain more fat because your energy expenditure is down. The fat is less insulin sensitive. And as you can see, it's a cycle that results in metabolic disease.
What is sort of the normal standard treatment at menopause for a woman? Do most women, or do many women, go on hormone therapies? Are they basically just taking extra estradiol to compensate for what was lost? What does that actually look like?
Well, it's a great question. It's a great question with kind of where we are in the world right now, because I think, historically, since the 90s, when there were the Women's Health Initiative trials, where to this day, the largest clinical trials only on women, and we spent more money on those trials than any other trial specifically for women in history. And anyway, there were problems with those studies, and it resulted in a shift in clinical practice. And so for decades, women really haven't been even thinking about hormone therapy very much. Some clinicians are prescribing it. The protocol has been lowest dose for as little time possible, only in instances where women are really suffering from symptoms of menopause. However, recently, I think things are changing, and I think more and more women are thinking about hormone therapy as being part of the menu of options of things that might help protect them as they go through menopause, in particular on the metabolic side of things. I think estrogen is critical for bone density. That's one of the major, I think, things that are that's really important. I haven't talked about it at all. It's fat cells are important, but bone, and it's probably the number one role of estrogen is bone health. And women that go through menopause have an increase in osteoporosis risk because of the lack of estrogen. So I think hormone therapy could be a good strategy for many women. And the FDA recently removed the black box label on hormone replacement therapy because of the just more research that has been done of late, and kind of revisiting those initial trials that were conducted in the 90s, and kind of backpedaling a little bit on the caution that was voiced then in terms of breast cancer risk and some other things.
Got it. So yeah, obviously there's, estrogen signaling ties into certain forms of breast cancer. So it's not the norm today that menopause will then trigger some kind of hormone replacement therapy. That is happening more, but that's certainly not the norm.
That's right.
Got it. And what are some of the main symptoms of menopause, and do we understand them in terms of the underlying hormonal changes?
Yeah, I think we don't understand them. I think there's so much more we need to learn about menopause. I think, again, we're starting to study it more and more in terms of in terms of kind of the symptomology. What you hear most about is hot flashes. Like that is one. Right. Vasomotor symptoms. That's one of the first physical manifestations of the drop in estrogen that happens. And we really do know it's sex hormone driven, because if you take a premenopausal woman and she has a hysterectomy or oophorectomy, she will experience those same symptoms. Night sweats are the major thing. But then there are other things like brain fog and joint pain. And there's all these symptoms. I mean, as you might imagine, because estrogen is affecting every cell in the body, it's doing lots of stuff. There's going to be a whole manifestation of things that happen, and it's not going to be the same for every female that goes through it. But hot flashes, there's cognitive changes. Sleep is a big thing that changes. Women suffer from more insomnia. A variety of symptoms that are noticeable. And then I think even more important are the symptoms that aren't really noticeable, like the accumulation of belly fat and the reduction in the bone density. And on the metabolic side of things, a reduction in energy expenditure. My lab is particularly interested in the energy expenditure that is voluntary, meaning motivation for physical activity, which I think actually goes down in menopause. Yeah. And we see that same thing happen in rodents when we remove their ovaries. It's really really astounding to see in the cage, the mice, within 2 weeks of not having ovarian estrogens reduce their physical activity.
Yeah. Well, I was actually just about to ask you, I would assume mice and rodents don't naturally go through menopause because they just don't live long enough. So I was going to ask you, like, well, it sounds like we probably can't model that, but maybe we can. You guys can remove the ovaries, and that maybe looks like menopause to some extent.
It is. It's imperfect. It's imperfect, but it's surgical removal of the ovaries. It's sort of like a hysterectomy in a female. So it's not perfect. It certainly doesn't replicate the perimenopausal transition. And it's abrupt. We just, remove the ovaries, but you do recapitulate many of the same things that you see in human females with menopause, in particular regarding the adipose tissue changes and metabolic changes, insulin resistance increases, inflammation increases. And what's really important in my lab, and what I'm particularly interested in, is the drop in physical activity. So there have been clinical studies to show that women going through menopause gain weight. We know this, they gain weight, and it's due to the drop in energy expenditure and not a change in what they're eating.
Yeah, that's interesting, because I was actually thinking about something you said earlier. So if estrogen tends to lead to a browning of adipose tissue, and that corresponds to an increase in mitochondria, that would imply, I would think, a change in basal metabolic rate.
And it absolutely does.
Let's be explicit about that for people. What is the effect here on basal metabolic rate? And can you just kind of describe what you just said in terms of basal metabolic rate and the number of mitochondria?
Yeah. So number of mitochondria is definitely going to predict basal metabolic rate for all the reasons that you described. Lean mass has a greater contribution to basal metabolic rate than does fat mass because of the mitochondria. In just the energy equivalent of keeping that type of tissue functional. So as women, when you reduce the mitochondrial density in white fat, for example, or mitochondrial activity or fat oxidation or any of these things that are kind of fostered by having more estrogen, all of those things are going to go down with estrogen loss, and that's going to contribute to a reduction in basal metabolic rate. Not to mention, again, as I mentioned earlier, the anabolic resistance and less lean mass, that is all together going to contribute to a reduction in basal metabolic rate. In addition to that, we think that there is a reduction in voluntary physical activity, which is driven by a reduction in motivation for physical activity, which is something that happens kind of neurologically.
Right.
So,
but even if a woman kept everything identical and was going through menopause and went through menopause, she ate the exact same number of calories, the exact same diet. She, despite any changes in motivation, brought herself to exercise and move in exactly the same way. You would still expect to see changes in fat tissue distribution. You would still expect to see a change in overall metabolic rate and weight in these things, because her cells themselves are changing in terms of their energetic capacity, let's say.
That's right. I think you can buffer some of it with what you've described, like diet and exercise, and maybe entering a little hormone replacement therapy into the menu of options. I think there are ways that we can kind of buffer some of what's going on, but you're absolutely right. I think aging in females is associated with a reduction in energy expenditure for the reasons that you described.
So the hormone therapies that some women get when they go through menopause, that's not the norm. We haven't been doing them forever. Do we know much there about the risks of those and how effective they are at correcting some of the symptoms and problems that come along with menopause?
Short answer is no, because we don't have the data yet. I think the data that are available show that yes, there's risk. I think there's risk with any type of pharmacological therapy. I think women who are sensitive to estrogen triggered cancers like breast cancer, uterine cancer, ovarian cancer, estrogen, would maybe not be a good idea in those women. It would be like it would have to be a conversation with the clinician in the case by case basis. But I think broadly speaking, my opinion on the read of the literature is that for a woman who is relatively healthy with no, increased risk of breast cancer based on her history, I think the benefits of doing hormone therapy outweigh the risks. But that's my opinion based on limited evidence that is available.
Got it. So the hormone therapies will mitigate some of the symptoms to some extent because they're coming largely from this drop in estradiol and this change in circulating hormones. There must be some risk. We know that certain breast cancers are sensitive and tied to estrogen signaling. So there might be some risk there. Obviously, you're going to have to do a risk benefit analysis with a physician, depending on, the individual and what all your history and risk factors say. And we just don't know really the long-term, quantitatively what the long-term risk profile is, because we really haven't been doing this for very long.
That's right. I think that's an accurate summary. And I think what people know less about is that I do think estrogen therapy or hormone therapy would play a role in mitigating the metabolic risk. And I think we've heard much less about the metabolic risk. We hear a lot about the cancer risk. We hear a lot about cardiovascular, stroke, blood clots, that side of things, which that's not my area. So I defer to those experts. But I think women hear much less about what's going on metabolically, and the data that are available, I think are clear in that it looks like there would be a net benefit metabolically.
Yeah. I mean, the strong expectation should be that estradiol replacement after menopause should lead to greater insulin sensitivity and less predisposition to visceral fat accumulation.
That's right.
Yeah. And obviously metabolic disease is becoming more and more common. So that's a serious consideration, I would think.
That's right.
Yeah. But just kind of tie the bow on some of this stuff. Estrogen, other hormones, very powerful. They act in all cells or nearly all cells. They change behavior through actions in the brain. They change basal metabolic rate by affecting mitochondria, stuff in fat tissue and elsewhere, affecting all sorts of things. So they are going to have powerful effects on what the body looks like, what the body does, how someone behaves, and just their personality, whether we're talking about estradiol as a replacement therapy, menopause, or used earlier in development. And that would be true for males and females, right? We're often talking about things in this discussion in a female-centric lens because they go through menopause and that stuff. But estrogen signaling is happening in males too, and it's going to have powerful effects throughout the body.
Absolutely.
What are some of the things, can you give us just a brief description of what are some of the things that you're working on in the lab today that are on the horizon that you'll think we'll get interesting answers to in the next year or two?
Yeah. So the thing I'm most excited about right now is this brain region called the nucleus accumbens. It is the region that we think about when we think about drug addiction and just motivation for anything, for food, for sex, for all the things that keep us alive. So I think it's a really fascinating brain region. And I started becoming interested in this brain region over a decade ago. Whereas I have a colleague, Frank Booth, who has been studying for years motivation for exercise. And he's done all these profiles in the brain and came to this brain region as being one that he sees correlated very strongly with motivation for physical activity. And one of the early studies that I did when I started at Mizzou was looking at the nucleus accumbens brain region in overectomized versus sham operated rats, and showed that that region is changing with estrogen loss in pretty incredible ways. And so our most recent study was one where we really wanted to look at what estrogen is doing in that brain region to affect behavior. So in terms of motivated running behavior, but this actually links to the fat too. And so in those most recent study, we exposed rats to estradiol direct to their nucleus accumbens. So we set them up with stereotaxic injections to the nucleus accumbens following overectomy. And we just published this work, where we showed that estrogen direct to the nucleus accumbens was sufficient to increase running behavior in rats. We didn't fully recover exercise levels to the pre-overectomy levels, but we did increase their levels. We did another study in mice where we did sort of a genetic manipulation in dopamine centric regions of the brain. So the nucleus accumbens is really the motivation center. So the dopamine signaling is kind of signature in that particular region. And so we manipulated an estrogen receptor. So we kind of tweaked estrogen signaling in the nucleus accumbens. And in that particular study, we didn't see effects on behavior, but I saw effects on adipose tissue. And so we saw an increase in browning of adipose tissue with a manipulation in that region of the brain.
Right.
And so I'm very excited about looking at the nucleus accumbens as sort of a mediator between behavior and exercise and adipose tissue physiology.
Yeah. And I think this just ties into what's easy to forget for some people, which is, right, the brain and the body are not, it's all one connected thing. One. And, they affect each other. So you're basically saying that estrogen signaling, if you manipulate it in the brain only, in one region, the nucleus accumbens, which is a region that's part of this dopaminergic mesolimbic reward system, the reward part of the brain, let's just say. If you change estrogen signaling in the brain, not systemically, not in the whole body, it can actually affect not just behavior and things like the motivation to do something akin to exercise, running on a wheel for a rodent, but it can change the fat tissue. So manipulation in the brain from estrogen is changing what the fat tissue looks like elsewhere in the body.
Yeah, isn't that wild?
Yeah.
But and I think it's, when you think about evolution and how important reward is in survival and how important freeing up energy resources from fat cells is for survival, I think it makes sense that there's a link there.
Right.
And we need exercise to get to find mates and to find food. And we need those things to be rewarding. And we need our adipose tissue to be sensitive to knowing when we need energy and we need to be storing energy. So I think it makes sense that those things are all connected, and we're excited to kind of start to tease out what those connections really look like.
Are there any final thoughts you want to leave people with, or anything you want to reiterate from our discussion today?
I think I talked about a lot of things. I think mostly, I think estrogen, I'm fascinated by estrogen. It's having so many roles in the brain and in the body. And there are so many overlapping functions of estrogen and exercise. I would say that, and we didn't talk a lot about exercise, but from the perspective of all of the things it's doing to mitochondria and metabolic health and just all the things, rewarding effects in the brain. Exercise is free of risks. Estrogen gets a little, dicey territory talking about, because it's very kind of polarizing and can be controversial. But exercise, I feel like, is something that is not very controversial, and there's no no side effects to it, and it's only going to do good things for you. So that my public health message would be exercise, embrace it, and Can I ask you one more question? Of course.
Okay. So it's going to be a little bit more of a big question. Based on everything that you've told me, right? It's just estrogen affects lots of stuff. It goes to the fat tissue. It goes to the brain. It orchestrates changes all over the place. It affects behavior. It affects metabolism. It affects mitochondria. It affects our propensity to act and exercise and do all of these things. We've been giving a lot of estrogen to a lot of people for a long time. Have we perhaps underestimated a little bit how powerful these manipulations are beyond their direct effect on reproductive things?
Oh, probably. I think the poignant point, yes, because estrogen is extremely powerful, and I think most of us don't really appreciate the extent to which it is affecting all systems of the body. So is it something we need to be thinking about more? I think probably. And we talked about like natural estrogens, you talk about endocrine disruptors and all of those things in the environment. I think those are things that definitely maybe haven't been considered enough. And we need to, reevaluate some of those and think about things a little bit more given that. Yeah.
All right. Well, this is fascinating stuff. I was excited to learn about your research, and I look forward to following along. So once again, thank you very much for your time.
Well, thank you. This was fun.
Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio, formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.